Ya, hari ini kita sungguh bersyukur karena kita kedatangan seorang uh, ahli di dalam bidang lingkungan, khususnya yang berhubungan pertanian dan peternakan. Uh, dan sebelum kita mulai, saya akan memperkenalkan Mr. John. Jadi, Mr. John adalah visiting lecturer atau dosen tamu yang berada di Undana selama 6 bulan dari Fulbright. Ya, jadi dia biaya Fulbright dan sudah berada di Undana dari bulan Februari sampai Agustus nanti. 1 semester di sini dan sudah melakukan perkuliahan di pasca dan beberapa uh, prodi lainnya. Dan hari ini kita akan mendengarkan Mr. John untuk menyampaikan beberapa hal terkait keahliannya di bidang soil nutrient soil nutrient stoichiometry in grassland in response to grazing and burning. Jadi beliau akan membawakan materi tentang nutrisi tanah, stoichiometri nutrisi tanah dan hubungannya dengan rumput dalam juga peternakan ya dan juga pembakaran. So let me introduce Mr. John. John Mischler is a associate professor at the Goshen College. Am I right? So yeah. He finished his Ph.D. in ecology from University of Colorado Boulder, 2014, and he also completed his master degree in geoscience and bachelor in geology, physics, and maths. So I think Mr. John is really a scientist, truly scientist, and he will share with us his expertise in uh, soil nutrient stoichiometry in grassland in response to grazing and burning, because that's one of his expertise. Mr. John also involved in some part of the world, for example, in Africa. So maybe you can share with us later on a little bit about what you're doing around the world. Now I'll hand it over to you, John. So your time is now.
Terima kasih Pak David. Selamat pagi. Maaf, saya sedang belajar bahasa Indonesia, tetapi saya hanya tahu beberapa kata saja. Speak bahasa English, and if anyone has questions or needs additional clarification on understanding parts of the discussion, please just let me know. You can just speak, and I'm happy to add clarification. I'm speaking English. Yeah. So I'm honored to be with you this morning. I'm very happy to be able to spend some time with you sharing both some of my work, but also a more general description of ecological and environmental stoichiometry. So the interaction between chemistry and ecology. I am a biogeochemist, and so I specialize in measuring and understanding how chemical elements, especially carbon, nitrogen, and phosphorus, how they move and determine the function of ecosystems, and also how human effects can change how these elements move through systems and how these systems function. So I will start with a general discussion about ecological and environmental nutrient stoichiometry, and then move into discussing one of my projects that I've been working on within grassland. Sorry. Okay. So, what is stoichiometry? So, I know all of you are familiar with stoichiometry, very basic and important term within chemistry. Basically, at the most simple description, it's just talking about the relative proportions of different elements, molecules within chemical reactions. So paying attention to the specific ratios. Now, when I'm talking about environmental or ecological stoichiometry, I'm moving into how these specific elements and the ratios between these elements are determining the functioning of ecological systems. And specifically, in ecological systems, three elements that are important for growth are carbon, nitrogen, and phosphorus. So carbon, nitrogen, and phosphorus are crucial for any living organism, and any living organism is made up of mostly carbon, nitrogen, phosphorus, oxygen. So carbon, nitrogen, and phosphorus are the nutrients that determine growth within ecological systems. And specifically, we pay attention to ratios between carbon, nitrogen, nitrogen, phosphorus, carbon, and phosphorus. We look at ratios because we are interested in understanding what is limiting limiting elements, limiting nutrients within ecological systems. So the ratios can help us understand what what elements are limiting within a certain ecological system. So what do I mean by limiting? So sometimes it's mostly nitrogen and phosphorus that are limiting within ecological systems when it comes to nutrients. But sometimes you can have a carbon limitation as well. But life requires ratios of carbon and nitrogen and phosphorus within specific ratios. So if a certain organism needs to maintain homeostasis, right? Homeostasis means just being able to stay operating at typical efficiency. So in order to maintain homeostasis, a particular organism needs to assimilate, needs to take in carbon, nitrogen, and phosphorus at specific ratios that will match its body stoichiometry. So oftentimes the food or the resource that the organism has access to does not perfectly match. In fact, almost always that resource does not typically match the ecological stoichiometry of the organisms. A simple example involves humans. You think about a meal that you might want to eat, right? So you have rice, which is very high in carbon, right? Starches. Then you have some fish, right? Which is high in nitrogen and phosphorus. And so we match, making a complete meal for ourselves, we try to match the stoichiometry of our bodies in order to provide what we need to achieve homeostasis. And in the similar way, organisms and ecosystems also are trying to assimilate carbon, nitrogen, and phosphorus within the proper ratios to enhance not only growth, but decomposition rates. So bacteria, microbes are taking in carbon, nitrogen, and phosphorus within specific ratios. And even biodiversity. So the idea that certain organisms may exist or won't exist within the ecosystem, or certain organisms may take over a particular ecosystem depending on the carbon, nitrogen, phosphorus stoichiometry of the food resources in that system. So when we're thinking about ecological stoichiometry or environmental stoichiometry and carbon, nitrogen, and phosphorus, it's important to look at both the resource nutrient stoichiometry, but also the organism nutrient stoichiometry. So, for example, if the resource is a lot higher in nitrogen, let's say the organism is using a protein-rich food source, then nitrogen will be excreted into the system because the organism is trying to remove or get rid of the extra nitrogen that it does not need. So I'll give you an example from some past research that I've done. So one research system that I've been working with are trematode parasites. So they're worm parasites that have multiple hosts and multiple life stages, but the first life stage is they're living within an aquatic snail. And so these snails have a particular homeostatic carbon, nitrogen, and phosphorus ratio. But when they become infected with parasites, then their their total body, including the parasite, stoichiometric ratio changes. And what that means is not only do they need more carbon because the parasites are carbon rich, stoichiometrically speaking, compared to the snail alone, but also they need more phosphorus because an important concept within ecological stoichiometry is that the smaller the organism becomes, it often becomes a higher percentage of phosphorus. And you can look at these elements, the ratio of these elements, and actually relate it back to cellular structures. And so specifically mitochondria have high amounts of phosphorus. And so the higher the density of mitochondria within an organism, the more phosphorus that organism will need compared to nitrogen and carbon. And so specifically organisms that have a very fast growth rate, and so this usually includes reproductive tissue, often requires more phosphorus. So in this way, this is one example of how ecological stoichiometry explains what we are observing in the natural world. So we see a snail alone, and it's consuming food and excreting according to its own nutrient ratios, its own ecological stoichiometry. Once that snail becomes infected with a parasite, then we see the excretion patterns of that snail change so that the snail is excreting more nitrogen back into the environment than it was before that it was infected. And that's because the parasite infecting the snail changes the snail's homeostasis and requires a different stoichiometry, a different nutrient ratio than what it did when the snail was not infected. So ecological stoichiometry allows us to not only describe ecosystems, but be able to make predictions and understand how these systems might function within different contexts. And it's also important. So humans, we have greatly altered, we have changed nutrient cycling on Earth. So when thinking of human activities, so fertilizer, runoff, uh, fossil fuels create nitrogen compounds that go into the air. Um, we have increased the content of carbon dioxide in the atmosphere, so changing how much carbon is available to plants. We have, we change by mining phosphorus from the ground, or even keeping farm animals where their manure is going to be depositing across the ground. We're changing the nutrient availability within ecosystems. And there's an entire area of research doing what's called nutrient additions, where we want to see how ecosystems are responding to human caused nutrient additions. And so we actually add nutrients into certain ecosystems to see if the makeup of organisms is going to change, so if the biodiversity is going to change or decomposition rates are going to change. So ecological stoichiometry, carbon, nitrogen, and phosphorus ratios, is a really important and useful tool within ecology. So the one of the most important sort of key concepts when talking about ecological stoichiometry, where it all started, is the Redfield ratio. So the Redfield ratio, back in 1934, Alfred Redfield just found that carbon, nitrogen, and phosphorus were at a fairly constant ratio within marine phytoplankton in ocean waters. So 106, and this is on an atomic basis. So 106 atoms of carbon to 16 atoms of nitrogen to 1 atom of phosphorus. So this was the first time that humans started to measure stoichiometric nutrient ratios and start to see patterns, right? So Redfield was interested in trying to understand how phytoplankton, lots of different species in lots of different places in the world, why did they all have this similar carbon, nitrogen, and phosphorus ratio, 106, 16 to 1? And so it ends up, so the answer is that this ratio indicates that nitrogen and phosphorus are often limiting within ocean systems, within marine systems, and that these phytoplankton were all relatively similar in their need for carbon relative to nitrogen relative to phosphorus to maintain homeostasis. So this was the first time that we started to see broad patterns in ecosystems that indicate that not only within a species, but even within functional groups like marine phytoplankton, we can end up with these ratios that describe the need that organisms have for carbon relative to nitrogen relative to phosphorus. So since then, we have discovered that carbon, nitrogen, and phosphorus ratios are useful not just for phytoplankton and not just in the oceans, but useful in many, many different contexts. So many different organisms that, like the example that I mentioned about the uh, the snails and the parasites. So many different organisms across many different species, many different functional groups can have different stoichiometric ratios to maintain homeostasis. And I mentioned smaller organisms, larger organisms, reproductive tissue. So even within an organism, right, different life stages or even different parts of an organism can have different stoichiometric ratios depending on its function. Like reproductive tissues have higher phosphorus compared to nitrogen and carbon, whereas muscle tissue, proteins, has higher nitrogen compared to phosphorus and carbon. And then fats, right, as you would expect, have higher carbon compared to nitrogen and phosphorus. And then you average these all together and you get the carbon, nitrogen, phosphorus ratio of the entire organism. So we're specifically interested in how these ratios can help us understand ecosystems. So we use these to understand nutrient cycling within systems. So how are resources or food moving through systems, whether you're talking about a small sort of pond, small water body, or you're talking about the entire earth, you can understand how nutrients cycle in these ecological systems using ecological stoichiometry. Also, understanding concepts like eutrophication. So, eutrophication involves adding nutrients to freshwater and saltwater systems and increasing the productivity of those systems to an extent that there's too much there's too much algae, which then creates too many consumers of the algae, which then pulls oxygen out of the water column and leads to oxygen depletion in the system. And whether or not you get eutrophication by adding nitrogen or phosphorus depends on the stoichiometric ratio of that system. Some systems might eutrophie by adding nitrogen. Some systems might eutrophie by adding phosphorus, and some systems might eutrophie by adding both nitrogen and phosphorus together in different ratios. So that's what I was referring to when I mentioned limiting nutrients. So if nitrogen is limiting, this will, we will see this within the stoichiometry, or phosphorus is limiting, we'll see that in the stoichiometry. And so we know that the system is particularly sensitive to adding nitrogen or phosphorus. So if we're talking about restoration ecology, like restoring mangroves, for example, if the site has not had mangroves for a while, then the soils might not have a proper carbon, nitrogen, phosphorus ratio in order to have a successful restoration. And so we may need to actually adjust the carbon, nitrogen, phosphorus stoichiometry. And also, for example, when thinking about climate change and how carbon might be sequestered whether it's blue carbon being sequestered in the ocean or carbon being sequestered on land within soils. Understanding the carbon, nitrogen, phosphorus ratios, especially for decomposition, can change our understanding of how quickly carbon can be sequestered in these systems. So, in summary, generally speaking about environmental and ecological nutrient stoichiometry, so we have this quantitative framework, right, that we can actually make predictions based on the stoichiometry of organisms that make up an ecosystem. We can make predictions on how um, both environmental nutrient stoichiometry can affect the organisms, but also how these organisms can then feed back and affect the ecological stoichiometry of the environment. And we find that these stoichiometric principles work across scales, right? So working even within organisms, within the bodies of one organism, or looking at global biogeochemical cycling, these stoichiometric principles are similar across all scales. And one of the key observations is that it's not just how much, it's not just how much nutrients you have, but the ratios between the nutrients, specifically carbon, nitrogen, and phosphorus ratios. Okay, so now that we've discussed a bit about what ecological stoichiometry is, I want to provide a little bit of information, a little bit of context for a project that I've been working on, and also some interest in a project that I'd like to start here in NTT. So we're working both in Kenya and in the United States. So this is where I'm living in the United States, and then this is where we're working in Kenya. And we're looking at one of the things we're looking at is the stoichiometry of carbon, nitrogen, and phosphorus within grassland systems and how this might be affected by two very common management practices within these systems, which is biomass burning, so burning grasslands, and grazing, right? And so all of you are familiar with these management practices because they're also very common here in NTT, using fire as a management practice in grasslands and using grazing as a management practice in grasslands. So what's interesting about these are two very different contexts, right? Indiana, where I'm living now, humans, moved into North America and started using fire, 6,000 years ago. Historically, grazers were an important part of this landscape, but most grasslands in North America have been converted, especially in the United States. Most grasslands have been converted to agriculture. So we've actually lost most of our grasslands. And so in Indiana, in the United States, our context is more about restoration, trying to restore some of these grasslands and be able to enhance carbon dioxide sequestration within these grasslands. Now, the Kenyan context is very different. So much longer history of human habitation, humans living in Kenya. And there's still this history, this evolutionary history of grazers, but we have a much larger percentage of native grasslands that are still in place in Kenya. They continue to be converted to agriculture. And, here in NTT, we see similar things, right? The grasslands, some of them are being converted to agriculture. So, but Kenya still has many intact grasslands. So now in Kenya, it's shifted more to this idea of protecting the grasslands are that are in Kenya and to avoid them being converted. And so similar to similar to Sumba, for example, where there are large sort of grasslands and savanna areas, that drought can really strongly affect carbon dynamics for any certain year within these grasslands because like NTT, Kenya has a very distinct wet season and dry season, a monsoonal climate, and the dry season can be quite long, but it varies. Which, I was surprised yesterday as I was going to the graduation ceremony that we were getting rain here in Timor. So we know that sometimes the monsoonal climate can lead to really hard to predict dynamics. So, a few points that I want to cover is so very basic points to sort of lay out the foundation of what I'm talking about here is that warming on planet Earth, right, is due both to CO2 release by human activities, but also changes in habitats that can reduce a habitat's ability to sequester carbon, right? And loss of native grasslands, so where we have lost large amounts of native grasslands, this actually contributes to the amount of carbon dioxide in the atmosphere because you're taking carbon from the soils in these native intact grasslands, and when you turn them into agricultural areas, you're releasing that carbon into the atmosphere. And so because of that, so grasslands, and so often we hear about forests, right? Forests. Trees store carbon, which is true. But there's some of the most recent research has shown that grasslands could potentially store even more carbon than forests per area because grasslands put carbon into the soil, whereas forests, most of their carbon is held above ground in the woody biomass. Grasslands store massive amounts of carbon in soil. And fire and grazing both are likely to play huge roles in how carbon is stored in the soil. And because fire and grazing are both human, used extensively in human ecological systems, we need to understand how fire and grazing might affect the nutrient stoichiometry of a certain system. So, as I mentioned, and I'm gonna move through some of these slides pretty quickly. So, as I mentioned before, this idea that carbon dioxide, right, is not just about fossil fuel release, but it's also about how humans have been managing the. And so specifically, if you take a grassland and you remove the grass, you work up the soil, and you convert it to planting crops, the when you open that soil, you increase oxygen to the microbial communities that are living in that soil, and then they can undergo aerobic decomposition. So you've basically provided oxygen as the missing element so that these aerobic microbes can start to metabolize faster and faster, and they convert just like when we eat food and we breathe out carbon dioxide. Microbes also release carbon dioxide when they're using resources, and so they're converting organic carbon within the soil to carbon dioxide that's in the atmosphere. And when I'm talking about the United States, we have converted much of our grasslands into agriculture, and through this mechanism that I mentioned previously, we have released large amounts of carbon into the atmosphere. And so I'll mention a little bit about my background. And so this aerial image, this is where I was a child. So I grew up in this place. And you can see that it's surrounded by agricultural fields. So it's mainly corn and soybeans. And it was all grassland before humans converted it to agriculture. And so it's a very big landscape change. And my grandfather, he would go out with his grandfather and he told stories. This is called tiling. So these were wet grasslands. And so before they could be converted for agriculture, we humans needed to dig trenches and put in clay pipes to drain the water from the land. And so my grandfather would go with his grandfather, and the very last areas of native grassland where I lived were converted into agriculture by my grandfather and his grandfather. And so this is me as a child with my father. And so we lived and worked on that land, those farm fields that were converted from grassland and converted into farmland. And now this is me with my family. And so now I'm working to restore some of these grasslands back to the landscape. So I've gone in my family and my ancestors, I've gone from we were people who were removing grasslands and then utilizing these for agriculture, and now we're coming back and we're actually trying to restore some of these grasslands. So when thinking about grasslands and how they function, we think about plants as a carbon pump, right? So plants respire, right? And they photosynthesize. We also respire, but humans can't photosynthesize. So that's plants' power. And so plants are able to take carbon from the atmosphere, fix it into organic material. And so plants are able to create their own food, right? So these plants not only create starches and sugars for them to respire, but they also create something called root exudates. Root exudates are carbon-rich materials that they release in the rhizosphere, the root area. And they do this in order to farm, to grow microbial communities that assist the plant, right? So plants can plants are able to access nutrients within the soil by releasing these root exudates and farming or raising a microbial community within the rhizosphere, within their roots. And so plants fix way much more carbon than is required for their respiration in order to provide their soil microbes with this carbon. And then so they have carbon that's held within the above ground biomass, within below ground biomass, but there's also carbon that becomes fixed within the subsoil, right, by these microbes who are living and dying. So we call that necromass, microbial necromass, the dead microbes. So, and just to give you an idea of what's happened in the United States, so all of this green is where tall grass prairie, these are grasses that are about as tall as a person. Tall grass prairie used to exist, and almost all of this is gone now. Almost all of this has been converted to farmland. So this is where I grew up as a child, and this is where I live now. And so large amounts of land conversion. So now let's think about how fire and grazing, right? How fire and grazing might affect nutrient cycling and carbon sequestration within these systems. So when we talk about grasslands, I mentioned forests, the carbon is in the above ground, right? The carbon is in the above ground. Whereas in grasslands, so much of the carbon, both in root biomass, but also these root exudates, is below ground. So these large root systems are really, really important when thinking about the nutrient stoichiometry of the system. And specifically, what this project aims to clarify is understanding how carbon is sequestered within grassland systems. So our first idea, right, is this sort of leaf litter paradigm that you think about grasses, they grow during the wet season, and during the dry season, they die and then they fall to the ground and they start to decompose at the surface, right? And that is one way that carbon is incorporated into the soil, is sort of the above ground biomass decomposing, sort of being worked into the surface. But we're becoming more and more aware of the importance of this below ground and microbial paradigm, this idea that not only do you have roots, right? You have these this below ground biomass, but you also have these exudates, right? These sugars that basically cultivate microbial communities. And so the idea here is that not only are we looking at just what's happening above the ground, but we want to know how fire and grazing affects these below ground processes in order to change the rate of carbon sequestration. And then when it comes to fire, we also are interested in pyrogenic carbon. So when a fire comes through and burns a grassland, you'll see bits of charcoal and ash on the ground after the fire has come through, and that's pyrogenic carbon. And that tends to be very hard to break down. And so it becomes a really stable form of carbon sequestered in the landscape. So this diagram, and we'll be looking at B here, the orange arrows, this is emphasizing the below ground paradigm, whereas the gray arrows are emphasizing the above ground paradigm. And so everything that I was describing earlier, you can see represented within this figure, this idea that not only do you have roots, right, below ground roots, contributing to the below ground biomass, but you also have the bacteria and other microbes that are dying within the soil, creating necromass and also creating carbon below ground. So when thinking about the rate, right, when thinking about the rate at which bacteria are um, are sort of grown and turned into necromass. So the rate of carbon sequestration by this microbial process, soil stoichiometry, carbon, nitrogen, phosphorus stoichiometry is a huge limiting factor on how quickly this process can move forward. And so root exudates, which are carbon rich, actually provide the catalyst to increase the rate of carbon sequestration within this microbial framework. Similarly, the rate at which above ground biomass becomes incorporated from the surface into the soil depends on the carbon, nitrogen, phosphorus ratio of that above ground biomass, because the decomposers, right, the organisms that are going to break that organic material down and move it into the soil, they also have a carbon, nitrogen, phosphorus ratio to maintain homeostasis. And so if the litter is too carbon rich, then it'll be very, very difficult to break down. And you see this, some of the most carbon rich grasses or carbon rich part of the grasses can actually sit on the soil surface for a long time because it's too much carbon compared to nitrogen and phosphorus. And so the decomposers cannot break it down very quickly. Whereas if it's a nitrogen rich material, so there are plants, legumes that fix nitrogen from the atmosphere, so they tend to be more nitrogen rich. So Lamtoro, and Gliricidia, the trees, they're nitrogen fixing trees that grow commonly in NTT. These, if you were to put these down onto the soil surface, they would decompose very quickly because they have much more nitrogen, and so stoichiometrically, the decomposition is able to move faster and more completely. So when thinking about, and I'm just going to introduce you very briefly to this research project, so we are really interested in using these concepts of ecological nutrient stoichiometry to understand how fire and grazing influence carbon sequestration in these soils. And so I'm going to flip through a few slides really quickly, and I'm just going to show you examples of how we're trying to address this question within our research. So we're looking at two different frequencies of burning, burned every year and burned every 3 years. And our control is not being burned. And then we're looking at one level of grazing, which is it's either grazed, and then our control is not being grazed. And so when we put all of these treatments together, we have 6 different sets of treatments within each replicate, and then we have 7 replicates. And so these are our 2 sites in Indiana. And you can see photos of what these sites, they started as farm fields, and then we restored them to prairies. So you can see what that process looks like over time. And we've been able to mentor many different students looking at both the effects of grazing, but also burning. And so we burn in March. So this is just after the cold season, after the snow is gone and it's dry enough, and we burn our grasslands. And then this is our site in Kenya. And so the site in Kenya is a little different in that we are still looking at the effects of burning and grazing, but we have more animals than just cattle. We also have elephants and buffalo and giraffe, and yeah, so in Kenya we have exclosures, right? So fences that try to exclude different animals so we can see, okay, what if it's just cattle? What if it's cattle and large wild animals? What if it's cattle and large wild animals and small wild animals? What if So we look at all these different combinations. And here's an example of burning in Kenya, which you can see the grasses are a lot shorter. So we are interested in quantifying different aspects of the system in order to get at this question of how burning and grazing affects carbon sequestration rates in these grasslands. So there's below ground net primary productivity, right? So we're looking at the root biomass and how quickly they grow and how quickly some of the roots are being decomposed into the soil. Above ground net primary productivity, right? So that's all the grass leaves and the litter. And then we're also interested in soil and carbon and nitrogen, right? And as part of this, understanding how the microbes, understanding the diversity in the biomass. So specifically the diversity helps us understand the stoichiometric values of the actual organisms that are doing um, that are taking the exudates in the soil. And so that helps us to understand how the stoichiometry of the original resource might be affecting the rate of carbon sequestration, right? And also the diversity in the types of different plants. Again, that's going to affect the ecological stoichiometry of the litter, which then affects the rate at which this litter is incorporated into the soil. So very briefly, we use these things called mini rhizotrons, which are clear plastic tubes that allow us to actually look at the roots in the subsurface, and we have a camera that measures them so we can measure them over time. We also are looking at the above ground biomass. So we can take quadrats and clip off the quadrats to measure the biomass of the grasses. And then this is how we sample soils. So we sample both surface and subsurface soils in order to measure the carbon nitrogen ratios as well as microbial biomass and diversity. And then we also want to know plant composition, right? Which plants are there, and we use something called a pin frame that's put on a quadrat, which is basically just a way to evaluate the different types of plants that we see in the system. So I'm gonna actually move through these results for now. So we do have data, but it's preliminary data at this point, and so we are going to continue to collect data over the years to evaluate nutrient stoichiometry. And so something I wanted to mention here at the end of this talk is some of the stuff that I'm interested in here in NTT. So this is the Amarasi system here in Timor. So and this is feeding cattle lamtoro and other plants. So lamtoro is a nitrogen-rich plant. It's a legume. And so part of what's interesting about these systems, right? And this whole hillside in Amarasi is lamtoro, almost all lamtoro. So lamtoro is from South America. And so you have this huge conversion of this was probably savanna before, and now it's very thick with lamtoro. So how does this grazing system, the cattle rearing system, change the functioning of the ecosystem when you include so many nitrogen fixing plants, right? So completely changed nitrogen fixation rates here in Timor. And also in Sumba, so Sumba has large open grasslands, and possibly seeing this increasing length of the dry season. So being able to evaluate fire frequency and intensity and grazing and what kinds of effects these are having on soil function in Sumba. to understand how we can optimize management practices to be able to increase the productivity of the grasslands in Sumba, right, is another interesting story. So with that, I'll pause here and I'm happy for any questions. Thank you very much.
Thank you very much, Dr. John. This is a very, very interesting topic. So now we can see actually as especially as we are as a chemist, the real life of stoichiometry that affect our lives. Yeah. Um, is there any question?
Yes, I do have questions, Pak David.
Okay, go ahead, please.
Yeah. Okay. Hello, Pak John. Good morning. It's nice to see you and finally talk to you. I've heard
You
about you.
are you Sinche? Sorry, your name is
Yeah.
just
Yeah.
okay.
Yeah.
Go ahead,
Yeah.
Sinche.
I'm
Yeah.
Sinche. Yeah. Okay. Actually, I have two questions, Pak John. First is I'm wondering how you measure the content of carbon, nitrogen, and phosphorus. The ratio. I mean, so for example, uh you measure the content or the ratio of these atoms, carbon, nitrogen, and phosphorus in a systems. So what the method that you use in your project?
Yes,
And
it's a good, and I'm sorry, I can answer this first question.
okay,
What,
yes, please.
so for carbon and nitrogen, we use what's called an elemental analyzer. And what the elemental analyzer does is you take your sample, it could be soil, it could be leaf biomass, it could be root biomass,
Mm-hmm.
and you dry it
Mm-hmm.
and you weigh it so you know exactly how many milligrams of material you have. And then you wrap it in a tin capsule and you put it in the elemental analyzer.
Mm.
The elemental analyzer completely there's a furnace inside that completely burns the sample, so it completely oxidizes the sample.
Oh.
And then it measures the carbon and nitrogen gases
Mm-hmm.
coming off of that oxidation. And so then you can convert those, since you know the biomass of your original sample, then you can convert those back
Mm-hmm.
to percentage of carbon and percentage of nitrogen. But
Okay.
so with phosphorus, there's a few different methods to measure phosphorus. The one that we have been using is the colorimetric method. So spectrophotometers, I know there are spectrophotometers here at Undana.
Yeah.
And so all we do is we mix up reagents that so we have to digest. So when you have a solid sample, you have to digest it so that you can get all of the material dissolved within an
Mm-hmm.
aqueous solution. So we digest it with some, with it's a sort of a mixture of acids and other chemicals. And then we take this solution and we add the colorimetric reagents. And so simply the darker the color, right, the more phosphorus is in the sample. And
Mm-hmm.
so I use a 96 well plate spectrophotometer. And so we just measure the darkness of the color. Who's just using a spectrophotometer and use a standard curve in order to measure phosphorus.
Mm-hmm. Mm-hmm. Okay. Okay. Thank you very much, Pak John. I have
Yeah.
another questions.
Yes, please.
Yeah. The next question is you have mentioned about a fire and the graceland. Um, I don't actually understand the relationship about those two. So maybe you can explain more about that, more detail about that, and what is your conclusion? Whether the fire is useful for for nutrient the soil or
Yes.
or what? Yes,
Yes,
please.
that's an excellent question. Thank you. So the mechanisms behind uh, fire and grazing and how they ecologically change a system. So when we're looking at grasslands, fire, right, and grasses have evolved to thrive in systems where there's frequent fires. And grasses have done this by actually moving the growth, like where the leaves grow from, are actually below the soil surface. So
Yeah.
they're able to survive fire and grow again. So
Mhm.
fire frequency, if there's no fire, if the grassland has no fire, then the above ground biomass grows too much, no fire, no grazing. It grows too much and it covers the dead material covers the soil surface. We call that thatch. And
Mhm.
it can actually result in reduced growth because the material is piling up, right? Now, if you have too much fire, so if you're burning every year, year after year
Mhm.
after year, which commonly happens,
Yes.
because some of the fires are not controlled, and even people who are grazing cattle will burn the grass so that new fresh grass will grow. And so when you're burning too much, then the grass is putting so much energy into regrowing, and it's pulling energy from the roots.
Mm.
And if you're burning too much, then the productivity of the grass is also reduced.
Mhm.
And so the idea is you want to be somewhere in between where your burning is
Mhm.
necessary to keep grasslands healthy, but you want to burn not so often that you're reducing the ability of the roots to regenerate the plant. And
Mhm.
so, similarly with grazing, if you graze too much, then, and especially because grazing has a different effect than just mowing.
Mhm.
So, and cattle graze different plants with different preferences. So, cattle are actually affecting the biodiversity of grasslands because they eat the most delicious plants most frequently.
Mhm.
And then if you are grazing too much, then the plant again is trying to regenerate from the roots. And if you're grazing too frequently, then the roots are not able to regenerate the plant.
Mhm.
But we found with intermediate, so this is the literature, with intermediate grazing and intermediate burning, you actually increase productivity and you increase the rate of carbon being sequestered into the soil sometimes. And so this project, there's a real lack of studies that look at both fire and grazing together. So this project is designed to address both, which fire and grazing often occur together in human managed systems. So it makes sense to look at both. Does this answer
Mhm.
your?
Yeah.
Okay.
Yeah. Yeah. Thank you, Pak John. And next, what is the ideal repetition time of grazing of a land?
That's an excellent question, and we can only understand that within a very specific context, right? So
Yeah.
in Timor, it may be different than in Sumba, which might be different than in Java, because rainfall can be different, right? The dry seasons
Right.
are much than the soil nutrients can be very different, even if you're talking about different breeds of cattle. And so the revisit, um, the revisit time is really different depending on the system and even the season, right? So it's going to be very different if you're grazing in January than if you're grazing in October. Yeah.
Mm-hmm.
So I can't tell you a number because the number will be very different.
Oh, yeah, 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 yeah. I'm wondering about this because here, our ancestor used this method actually to open the land for agriculture. So actually, they have applied this method to for agriculture.
Mm.
So thank you, Pak John, for your explanation.
Yeah,
Terima
excellent
kasih.
question. Sama-sama.
Yeah.
Ya, thank you Bu Sinche dan Pak John untuk jawabannya. Ada pertanyaan dari Bu Seri. There is another question in the box from Ibu Seri asking whether the opening of new farming land by burning grass, is it okay or not? Yep. Yeah.
It's a very good question. And it very much depends on the context. So when we were mentioning about ancestors, right, and how they were practicing agriculture, they were doing shifting agriculture. So they would take all of the trees, everything within a small area, burn everything, which provides nutrients to the soil, and use that area for a few years to grow the crop. And then they would move to another spot and then another spot, and they would move all around Timor, yeah, doing this. And so they might not revisit they might not revisit a place for 30 years. You know, there would be long time between between burning, so the land would have time to regenerate. And there were less people. Yeah, there were less people. So, but now there are more people, and people are more settled, right? People are not moving large distances all the time. They're staying in one place. And so now it becomes harder to use that shifting cultivation method, because the revisit time now, I would think, is much shorter because people are not moving as far. And so it was a very good method when your ancestors were using it. But the new contexts that we have now, we have to think about ways to change the way that we are farming, because there are more people and we are not moving as much. And so it's possible, now we have access to plants that we can plant within some of these agricultural spots that can replace the nutrients more quickly. And so there are many excellent examples from here in Timor and other areas in NTT of planting. You can plant plants together. So as a simple example, right, you can plant coffee, right, underneath avocado or mango or palms, so that you are growing more crops within a small space. And so there are lots of good examples here in Timor about how people are adjusting to the new conditions that are fairly recent, right? After colonialization, right? After the Dutch came, things started to really change. So again, the answer to your question is it depends on how you use the burning methods, and with the current style of living, we are also needing to adjust our style of agriculture. Yeah.
Thank you so much, Mr. John. Okay, maybe another question from Dr. Fidel. Dr. Fidel is a Amarasan guy, so Maybe.
Ah,
Please, Pak Final.
Selamat pagi, Pak John.
hello.
Uh, well, thank you very much, Pak David, for moderating this very interesting talk. So, Dr. John, this is a very interesting talk, and it's been a while since I think we've been in a class where I was translating for you in an environmental degradation class. Yeah.
Yeah.
So my question was related to the Redfield ratio, the ratio between carbon, nitrogen, and phosphorus. So my question is basically how does the environment, particularly the aquatic or land environment, manage to keep the Redfield ratio, the balance between those three um, wah, ketiga unsur itu
Mhm.
fairly stable. Uh, Even with all even with all the everything that happens in the environment through the anthropogenic activities such as a runoff from the industry or from the agriculturals, and what help maintains that kind of ratio in the environment. Thank you.
Mhm. Yes, thank you very much. That's a very good question. So the Redfield ratio is the carbon, nitrogen, phosphorus ratio found in marine or ocean phytoplankton. And there's a concept within ecological stoichiometry, which is homeostatic flexibility, right? So you could think about it in terms of a community of organisms. So if we take the grassland example, that a grassland is not just one plant, right? It's many different species of grasses, but also different species of other types of plants as well. And so the community within a grassland has an amount of homeostatic flexibility, which means if there's different soils, right, then you're going to find, and there's different nutrient resources within the soils, then the community will shift so that the community as a whole will reach a stoichiometric ratio that's suitable for the soils that it finds itself. And so you'll find different biodiversity and different types of plants on different soils. And we see this commonly, right? We experience this in our everyday life. So the makeup of a different community can shift its stoichiometric ratios to match the resource. And this can also happen for some organisms. So some organisms are homeostatically rigid. So they can't really shift their body stoichiometry to adjust to a new or different resource. And so they are limited in their ability to adjust, whereas other organisms are more stoichiometrically flexible. And so they're able to adjust their body stoichiometry somewhat to the resources that are available. And so some organisms are stoichiometrically rigid, other organisms are more stoichiometrically flexible. And the key that sort of structures how this, how this manifests in ecosystems is that it costs energy to eat or use a resource that is mismatched to your stoichiometry. So if your resource is much higher in nitrogen than what you need, then your body needs to excrete that nitrogen, and it actually takes energy to form the compounds in your body to excrete that nitrogen. Additionally, if your food was low in nitrogen, then your body would start breaking down the muscles in your own body to add the nitrogen that is necessary in order to metabolize the resource. And so the understanding how either communities or single organisms are organized around this idea of ecological stoichiometry really just depends on the fact that if at all possible, organisms or communities want to use as little energy as possible in order to maintain homeostasis. And so they want to be as well matched to their resource as possible. And so that's, for example, that's why for a human, it's a lot less energy intensive for us to eat meat than if we were to eat really high carbon material, but we need to make sure the meat has fat in it, right? Because we want the carbon from the lipids and then the nitrogen and the phosphorus. So I hope that answered your question as to how stoichiometry becomes structured, or how ecosystems become structured around stoichiometry.
Thanks, Dr. John. Thank you.
Thank you so much. Is there any question from students? You can ask question in Indonesian. No problem. Any? No, okay. I will ask. I have a question, Mr. John. As Kupang land is pretty different with others, a little bit rocky, and I think rich in terms of calcium, magnesium, and carbonate, I guess. Do you think that makes a difference in terms of the balance between carbon, nitrogen, and phosphorus? And do you have any thought how to deal specifically with this kind of land? Thank you.
Yes, it's a really, really good question. So just thinking about Timor and Sumba compared to Java or even Flores. Yeah, I think Flores. Flores, has many volcanoes, whereas Timor and Sumba, it's all uplifted coral reef. And so the rocks from which the soil derived are very, very different. So volcanic rocks, like in Flores, contain a lot of nutrients, a lot of micronutrients and macronutrients. But the uplifted coral here in Timor and Sumba are fairly nutrient poor. And they also contain a lot of calcium carbonate, because that's the material that the coral reefs are composed of. And so they are very basic. So they have a lot of buffering capacity. So they're not leached as easily, but they are also fairly nutrient poor because they're coming from the coral reef. So, and then in addition, during colonialization, the resources were cleared from the land, so like taking away teak and sandalwood, for example. And in this context, right, where you have a short wet season and a long dry season, and you have steep hill slopes, it takes a long time to form soil. And once you remove the soil, it might not be able to come back. And so if you look at Sumba, for example, Sumba has the large grassland areas, and so historically there would have been many more trees in Sumba, but now that the soil has been reduced, it would be very difficult for trees to grow there now. And so in this case, when we're thinking about restoration, sometimes you cannot go back. You cannot go backwards, especially when it comes to soil formation, because it takes such a long time. So in so in places like Timor and Sumba, where you have these very shallow calcareous soils, uh, it really takes effort to match the plants that you're growing with the soils that you have. And so when it comes to agricultural production, uh, there are certain crops, and I defer to, people who have been studying the agricultural systems of Timor. There are certain crops that are going to grow more easily than others, and especially when we're talking about the long dry season, or even varieties of crops. Right here in Undana, we're working on improved varieties for the NTT region, and so some varieties can deal with the more nutrient-poor calcareous soils here than others can. So yeah, I think when it comes to restoration, we have to remember that you cannot always go back to what existed before colonialization, because especially because of the low rate of soil formation. And then when it comes to agricultural systems, we need to pay close attention to the systems that we currently have and match our crops, or develop new varieties to match our soil systems. Yeah,
Thank you so much, Mr. John.
thank you.
All right. Is there any more question? If no, then I think we are almost done now. So I think probably that's enough for us today. Thank you so much, Mr. John. Again, we really got rich insight today, especially about the chemistry and environment, farming, grazing, and so on. Thank you so much. Now I will give a time for Dr. Fidel to uh, give a closing remark on behalf of the uh, head of the department, chemistry department. Dr. Fidel.
Thank you. Terima kasih Pak David. Terima kasih. It's an honor for me to deliver the closing remarks to represent the coordinator of the chemistry study program, who is not present today with us. So on behalf of the study program, I would like to extend our heartfelt thanks to Dr. John Mischler for for sharing your expertise on today's enriching presentation, particularly about soil nutrient stoichiometry. So I believe the insight that you have shared with us today, with the faculties and the students, have deepened our understanding about nutrient interaction in soil systems and their wider ecological implications. And I particularly appreciate your willingness to speak very clear and at a very slow pace, which help us to follow your, particularly the students, to follow the ideas of your presentation. And lastly, thanks also to all the participants for your present and continuous enthusiasm for today's monthly research talk. We hope that today's session sparks further discussion and continuous learning for your future. And for now, I believe thank you for everyone. I think that's all from me. Thank you.
Thank you so much, Dr. Fidel. Now I think we we can take the picture. Probably Mesa can help us to take picture. Please open your camera if you can. Students, every one of you. Silakan Pak Mesak.
Satu, dua, tiga. Sekali lagi. Satu, dua, tiga. Terima kasih.
Terima kasih Pak Mesak. All right. I think we are at the end of the session today. Thank you again for the participants. Thank you, Mr. John. We hope that we can collaborate in the future. We are now your effort is so I think we are very close with Kevin. Thank you so much and see you again. Bye bye.
Terima
Terima
kasih.
kasih.